Welcome to The Explainer. Today, we're going to dive deep into a single word that holds two totally different but equally fascinating stories in the world of science, and that word is fish.
Now, when you hear that, you're probably thinking of something with gills and fins, right? And you wouldn't be wrong, but that's only half the story. As we're about to see, the answer to this question is way more complex than you might think, and it shows how one little word can unlock two very different and very important scientific worlds.
Okay, first up, let's talk about guarding our waters. We're going to start with the fish we all know and recognize, the animals that are such a critical part of our planet, swimming around in our lakes, rivers, and oceans.
To really get a handle on this, let's head over to Hungary, to the Veterinary Medical Research Institute. Now, the scientists here are focused on what's called fish pathology. They're basically fish doctors, looking after the populations in huge vital lakes like Lake Balaton. And their work is absolutely crucial for protecting not just the wildlife, but also the fish that end up on our plates.
And let me tell you, these fish are up against some serious threats. It's a whole barrage of dangers out there, from tiny microscopic parasites and viruses to really aggressive bacteria. Things with names like Aeromonas and Flaviobacterium can cause some truly devastating diseases. And those scientists we just mentioned, they're on the front lines, trying to figure out how to fight these invisible enemies.
So what's the end game here? Well, their mission is to use all sorts of advanced lab techniques to really understand how these diseases tick. The ultimate goal, of course, is to find ways to protect both the wild fish and the ones on fish farms. It's all about safeguarding our food supply and just keeping our natural waters healthy.
So that's one meaning of fish. It's tangible, it's alive, it's swimming around. But what if I told you there's a completely different fish in science, one that's just as important, but exists on a whole other level?
And this slide right here, this really gets to the heart of what we're talking about today. On the left, you've got the animal, the focus of all that ecological and veterinary research. And on the right, you've got the technique, a seriously powerful tool used in genetics. Two distinct worlds, one shared name.
So, what is this other fish? Well, it's an acronym, and it stands for fluorescence in situ hybridization. It's this incredible technique that lets scientists look right inside our cells and see our genetic code by literally painting specific parts of our DNA with light.
So lighting up our genes. How in the world does this actually work? I mean, how do scientists make parts of our DNA literally glow in the dark?
Well, the real magic here is all about specificity. Scientists create this little piece of DNA or RNA, they call it a probe, and they stick a fluorescent molecule to it. Think of it like a tiny glowing beacon. This probe is engineered to be like a guided missile. It's designed to seek out and stick to one and only one specific sequence in the entire vast landscape of our genome.
It's actually a beautiful three-step dance. First, you create your custom glowing probe. Second, you introduce it to the cells, and it just naturally finds and sticks to its genetic partner. That part is called hybridization. And finally, the really cool part, you look under a powerful microscope and see exactly where that little beacon has lit up, showing you the precise location of a gene.
Now, this is where it gets really powerful. This isn't just some neat trick for the lab. The fish technique is an absolute cornerstone of modern medicine.
I mean, just look at this list. The fish technique is an incredibly versatile workhorse. Doctors use it in prenatal screenings to check for genetic issues. Oncologists use it to diagnose specific cancers by hunting for gene mutations. And researchers use it just to figure out where certain genes live on our chromosomes.
Let's make this super concrete. Take Down syndrome, for instance, which is caused by having an extra copy of chromosome 21. By using a probe that only sticks to that specific chromosome, doctors can use fish to just count the glowing dots. If they see 3 bright dots instead of the usual 2, they can confirm a diagnosis quickly and with incredible accuracy. It's that simple and that powerful.
Okay, so this brings us to the final, and I think most crucial part of our explainer. We've looked at two totally separate worlds, the living, breathing fish in a lake and these glowing genetic probes in a lab. Now it's time to see where these two worlds collide.
So we have to ask the question that's kind of been hanging in the air this whole time. Can this microscopic tool, this fluorescence in situ hybridization, actually be used to solve the very real macroscopic problems that actual fish are facing?
And the answer is a huge, resounding yes. Here's the connection, plain as day. Remember those researchers in Hungary fighting off nasty bacterial pathogens like Aeromonas?
Well, it just so happens that one of the well-established uses of the fish, technique, is to rapidly and precisely identify specific species of bacteria. The problem finds its perfect solution.
You know, this is just a perfect example of the power of interdisciplinary science. You have this really sophisticated tool developed for human genetics, and it can be directly applied to solve huge challenges in veterinary science and ecology. It just goes to show you how a breakthrough in one area can create unexpected solutions in a completely different one.
And that leaves us with one last thing to think about. One word: fish, connected two very different fields in a really powerful way. It just makes you wonder, what other game-changing solutions are out there, hiding in plain sight, concealed only by a shared name in the vast language of science?